All right, can everyone hear me okay? As Dr. Anderson mentioned, I'm Andrew Hopkins. I'm a new postdoc working with the Kennedy Center. My background is in disease ecology and ecotoxicology. So today I'm going to be talking to you all about new techniques for the detection of avian influenza in wetlands. So to kind of give a real quick overview of influenzas for those who might not have a good background in them, influenzas are a very broad class of viruses. One of the main influenzas that we'll be dealing with in this presentation is avian influenza. And one thing to notice about the influenza is it's a viral RNA, or it's a viral disease caused by the RNA protein inside. So that's what they use to kind of hijack our cell's machinery and use it to duplicate. But what's important to notice is that on the outside of the protein lipid bilayer, you have these two different structures, the hemagglutinin and the neuraminidase. These kind of help to create the different strains that we're talking about when we're dealing with avian influenzas and other influenzas. So commonly, you'll hear people talk about like H5N2 or H7N something. This is what they're referring to. So there are different structures that can happen on the outside of these influenzas, and that kind of helps to determine the pathology of these viruses. So commonly when you're hearing people talk about avian influenza today, you'll hear people say things like low path or high path. Low pathology avian influenzas, so one time I'm going to try and look back here, are things like H7N3 and H9N2. So these are things that don't really cause death, they just kind of give like a flu-like symptom. But the thing is, these low path variants can mutate into high path variants through two different methods. One is antigenic shift and antigenic drift. Think of one is why we need a new flu vaccine every year, and the other one is two different strains happening in the same place at the same time. They share these different proteins, and you get really bad one like H5N1, which is the main strain we're currently dealing with right now. So a relative timeline of avian influenza, we kind of have had avian influenza classified since about the 1800s when it was called foul plague. But with recent advances and as we kept on getting better detection mechanisms, we went through and have now classified these as avian influenzas. We've been dealing with these for pretty much ever. The main one that we're dealing with right now is called H5N1, and in 2020, it was first classified coming from Southeast Asian countries, most likely due to some form of mixing event leading to the creation of H5N1. By 2021, it's already spread to the Netherlands. By 2021 to 2022, it's causing multiple outbreaks worldwide. And by 2022, we're seeing our first commercial case in the US. That's why your egg prices went up, because this thing spreads so incredibly fast. The best way to go through and deal with it and keep it from wiping out your entire flock is to cull it so it can't keep on spreading and go to more places. By late 2022, we're seeing sporadic mammalian cases. Just because this is a bird flu does not mean it sticks with birds. It does get in mammals, it does get in us, and we've already seen fatalities from it. But 2023 cases spreading even further, and by 2024, we actually see two unique mutations that happen in this pathogen. Normally, avian influenzas don't really go and transfer over to dairy cattle. This has happened twice with two different strains of avian influenzas, whereas now transmissible in dairy cattle, and dairy workers are getting infected from these dairy cattle, which is leading to more human transmission and spread. That's likely going underreported right now. A little bit of happy note to end the day with. So the major species of interest that we're dealing with are a lot of the primary research in avian influenza right now is focused on domestic species. So again, most people are usually thinking of chicken flocks. However, wild birds are highly susceptible to avian influenza. They just vary in their susceptibility. Some are very susceptible to the point where avian influenza gets on a flock, the entire flock dies, and you don't know avian influenza killed them until afterwards. Other ones are really good carriers of this pathogen. So things such as wild geese are really good carriers of this pathogen. We don't really test for them as much, but they don't really get as sick, and they can carry it all throughout their entire migration pathway. And as I mentioned, this is just a list of all the different animals that avian influenza can infect. Like I said, just because it's a bird flu doesn't mean it sticks with birds. It's commonly found in seals and dolphins, so it's found in marine mammals. It's found in paradomestic species such as horses, ferrets, dogs, and cats. In fact, one of the main models right now for testing aerosolized transmission of avian influenza is a ferret-based model. And also, like I said, it can get in humans. We've already seen one death and one home dose patient from it, at the end of 2024. So the traditional sampling methods of avian influenza are very hands-on and kind of after the fact. The main way we kind of characterize and saying that avian influenza is in place is if a mortality event occurs, you go and sample carcasses, and then you can say, yes, we had avian influenza, that's what killed them. Because sometimes you'll get random die-offs of birds, sometimes it's aflatoxin, sometimes it's environmental toxins, sometimes it's a whole myriad of things to the point where we don't even bother writing them all down, we just say sick bird syndrome because it's drooping eyes, drooping wings, snaring discharge, and things like that. Other ways you can do it are by swabbing game species that you catch by hand or by having hunters turn in their kills so we can sample them. However, this creates a large bias towards harvested and domestic species. Again, we have a lot of testing under domestic poultry production. We can definitely test the ones that people are hunting, but the species that you can't hunt and in areas where you can't hunt, you don't really have an idea what's going on with the avian influenza population or avian influenza circulating in those areas. So that brings me to the technique that I'm trying to use, and that's kind of becoming a bit more widely available. It's called eDNA, which is short for environmental DNA. It's commonly used in bacteria. It was originally developed in about the early 2000s, and it works really well in water systems. So essentially, you filter water and search for try and detect DNA. Think of it as anytime you enter water, you are shedding your DNA into the water, and so is every single species. So you're leaving your own DNA particles in those water, comes from your hair, your skin, your saliva, everything. When you are entering water, you're leaving your DNA traces everywhere. Think of that next time you go in the ocean or a pool. There's also, but if you're sick, you're also shedding these RNA particles. So DNA double-stranded, RNA single-stranded. So if you're sick, you're also shedding this pathogen. So if a species is sick in the environment, it is shedding these RNA particles in the environment. You can detect these in the environment. However, I mentioned eDNA has a large focus on DNA, so there's not that much work with it in RNA. And as far as I can tell, there's only one real study before mine that is kind of focused on work that's published working on it, detecting it in wetlands. So why use eDNA? First of all, it's incredibly quick and cost effective. You can go through and sample a large entire wetland with very minimal effort. In my sampling, I in total probably spent 2 hours at each of my wetland sites, filtered between 40 and 80 liters of water, and was able to go through and detect avian influenza. Spoiler alert, I found it. It can be highly sensitive depending on what you're looking for. If your eDNA sample is strong enough and you get a good enough pathogen, you can actually go through and see what exact strain you have circulating. So you can actually be able to say, yes, this is H5N1, this is low path, this high path. Remember, just because you have low path in a wetland doesn't mean you don't have a chance of high path because it can mutate. It's incredibly customizable. So pretty much the entire process works by comparing it to a known library. So if you can go through and have a large enough database of DNA or RNA variants, you can pretty much set up a search method for anything you can think of. And finally, it's non-invasive. You don't have to get near the ducks. You don't have to touch the ducks. You don't have to freak out the ducks. You don't have to worry about hunters going, like interrupting somebody's hunting. You can do this in places where you can't hunt, such as your local community pond or stormwater ponds or different things like that in highly residential areas. So the goals of this study were to develop and test the efficacy of RNA detection. Like I said, not a lot of studies have really focused on using an RNA-based detection of eDNA. Also refine this methodology for South Carolina wetlands. A lot of the eDNA work that has been done looking for viruses and viral RNA has been primarily done in wastewater. There's quite a difference between wastewater and like natural stormwater ponds and natural wetlands. And also to develop a baseline for occurrence to see if we can detect it and if our methodologies work. So as you've seen before, we have our map of the flyways of the United States. We have the Atlantic flyway, South Carolina, lots of coastal wetlands. Birds go through, migrate through here. So even if they're just stopping for a little bit, they're stopping in all these different ponds that we're working with. They're stopping in the rice impoundments that everybody's talking about, but I'm willing to bet you drive down 17, you're going to see a whole bunch of birds occasionally stopping in all the stormwater ponds or whenever there's an overflow, or you go to your apartment complex pond, they have geese in there. So there's a whole bunch of birds that use all these different wetlands as different points for migration. They live there, or they just stop there for just a little bit. So how did I do this? I selected several sample sites from different wetlands, excuse me, including Sandy Coastal, Bear Island, and Whitehall Plantation. Next, I used filtering technology, so I have a peristaltic pump and a filter that I bring out in the field with me. This is the new filter we got. We've been using a previous one, which you couldn't even open it, but this one, exactly like the one up there, pop open the filter, or pop open the sampling tube, and there's this tiny filter inside. This is about 0.2 microns of nitrocellulose filter, which means it's incredibly small pore size, which means as it's sucking the water through, it's also going to be pulling through or all the viral particles are going to be stuck on this, which we can then later elute and bring into the lab and get some interesting information. After we filter it, I store it on dry ice and then pretty much wait until I can go up and work with the Clemson people up on main campus, go through and filter out my RNA. Next, this is all the fun laboratory stuff, or I'm going to try and go over this as quick as possible because I know not everyone wants to hear the fun facts of how you go through and pull RNA out of water samples. Essentially, we wash the RNA off of the filters into a vial, and we extract it using mag bead washing. So these beads actually bind the RNA to it. We then put them against a metal surface that pretty much pulls the beads off to the side, allowing us to wash off everything else that's not RNA. Then we wash the RNA off the beads, and we're left with this pretty much purified sample that's about like maybe a couple microliters of RNA at the end from, again, filtering 80 liters of an entire wetland. Then we screen it for detection. Ottawa kind of already mentioned like different adjuvant technology and tape stations. So we screen it for detection with a tape station to pretty much say, yes, you were able to pull RNA off of this sample, or no, there is no RNA present in this sample. So at the end of the day, what did we find? First of all, this was an exploratory study. I'm still going through and trying to find funding for this and see if it was even possible before I went for a larger grant. We tested around 20 samples. 10 of these tested positive for RNA materials, and those 10 that tested positive for RNA materials were sent to the Diagnostic Center at Clemson University. From those 10, one did test positive for avian influenza. So by sampling 40 to 80 liters of water from entire wetlands, and I'm talking wetlands the size of the main ones at Bear Island and different things like that, because Bear Island is where we did find a positive sample. We were able to confirm the detection of avian influenza. So again, think about that. I'm pulling just 80 liters out of that entire wetland, and I'm confirming that, yes, you do have avian influenza circulating in that wetland at that moment in time. So essentially for an exploratory study, this is pretty good. So yeah, that's the peristaltic pump. There's me trying to get somewhere near the wetlands where I can actually sample things a little bit better. So the implications, this is an incredibly viable method of sampling. It does not require you to actually have to interact with birds at all. It doesn't cause any undue stress, and it can be done in areas where much of the other forms of sampling and traditional methods of sampling aren't really feasible. It requires minimal effort and invasiveness, and it's also incredibly useful in other systems. Like I said, we focused on wetlands, but this can be done at apartment complex ponds where, again, you can see like all these different geese and different waterfowl in there. It can be done in stormwater ponds. It can be done in marine environments. It can be done pretty much anywhere that you want to go through and sample. You can do some form of eDNA sampling. And just because I focused on water does not mean it's only done in water. You can also look through soil environments too and go through and extract eDNA in those situations. So with having our positive detection and showing that this is a viable technique, what does this mean for future work and research? First of all, I want to try and increase detection. I've been working with the bioinformatics facility at Clemson to go through and get a better filter, get better methodology, and get better preservation, because even though we only found one sample, I'm willing to bet we probably had a lot more hits than just one. Avian influenza everywhere, and if I can get better quality samples, I can even be able to tell if we have high path in those wetlands, which is a major concern and a major thing to be able to tell people, like, oh, yes, you have high path in this wetland, or you've had a species stop by recently that has high path. Furthermore, we can go through and refine methods of sampling. So with these positive detections, we start taking environmental data to be able to see what types of wetlands might be more susceptible to holding avian influenza for longer amounts of time, or we can combine other techniques with it. Oxy's drone surveys can be combined with my avian influenza to be able to see which species are in these wetlands, how abundant they are, and if we can try and find a correlation or create a model to predict the spread of avian influenza, or maybe find different species that may be better carriers or transmitters than the ones that we classically think of. We can include more environments. One thing that I've submitted a grant for is to actually go through and start testing this in stormwater ponds, which are a major fixture around different areas around here. And they can definitely hold so many different species and possibly have different levels of pathogen here. And even go through off-season sampling events when you're not dealing with the hunting season. You can go through and still track and see if this is in your pond and see if it's in the area. Further study outcomes using rare species detection. I know these are probably very rare species that you guys would find in your ponds. Again, I'm not a duck expert. I'm a disease person. But you can also, like, if somebody's saying, yeah, I swear I saw a harlequin duck or a cinnamon teal or whatever type of duck, and they really wanted to figure it out, we could actually go through and say, yeah, if we can have a library or have some form of genetic sample to compare it against, yes, this is possible that you might have seen it in your wetland. Can be used in species recovery. So as Maya was talking about with her research, in terms of the mallard genetics, if you guys build up a large enough database of the different mallard genetics, the different strains you have sampling, if I can get a strong enough eDNA sample, I can compare and see, yes, you have a predominant game farm species in this pond, or this is a bunch of hybrids, or you might actually be seeing wild mallards. We'll be using different conservation needs for that. And finally, it can be used in further pathogen screening. Avian influenza is not the only major pathogen that's circling right now, it's just one getting the most news. Further pathogen screening, so amphibians and reptiles are currently suffering through multiple different diseases on multiple different fronts, including tritiomycosis, Bacterium salamander bands, and ranavirus. Both these can pretty much wipe out amphibian and reptile populations. So methodology can be used to kind of screen for the test and presence of these different pathogens in different areas, helping you to better be able to protect species and study the development and spread of these pathogens across the United States into areas where they might not have gone before. With that, thank you for your time, and I'd be happy to answer any questions and comments.
Would you be taking multiple samples from, like you have a wetland, would you be taking multiple samples from that wetland, or would you?
Yes, I did take multiple samples from the wetland, so I took one in pretty much each, like, of the four main compass directions.
Sure.
So each wetland had about 4 samples taken from it.
Okay.
Yes, sir.
So was Bear Island the only location where you detected?
Yes, that was the only positive we got, but
Only
again,
positive,
so it's the only positive that we got for avian influenza, although like I kind of said, the previous filters we were using, these ones are a lot nicer. You can actually access the filter material itself. The other ones I was using, you can't really open it up at all. It's literally sealed plastic inside of it. So the elution you're getting from the filters might not have been as strong or as good as we possibly could have done. So this is something that's going to actually help me be able to get better sample recognition, because I'm willing to bet there were a lot more positives than the single one I got in these ponds, because even if it's not high patch, low patch is also circulating in many different ponds.
all hunter collected birds.
Sir?
All hunter collected birds.
This has nothing to do with hunters. This is all
No, but I mean, where did the sample come from? How
water.
oh, it's all water. Okay. Oh, okay. I knew they were. I think they had tested for avian influenza with hunter collected birds at Sankey Coastal.
Yeah, so this methodology would not need to use hunter collected
Okay,
birds
yeah.
to exit in the environment.
Okay. I don't know Jim, who was involved in that? USDA, that's right.
Yeah, I was gonna ask, have you, so Bear Island and Santee Coastal Reserve, kind of off of Mike's question here. There is USDA sampling routine monitoring we do every year, swab hunter harvested birds. From both sites, there were positive hunter harvested birds, and you looked at maybe the correlation or any comparisons between, have you looked at that data?
No, not yet.
Okay. If you're interested in it, I can connect you with appropriately.
Yeah, that'd be great. Thank you. Yes, sir.
Well, this is neat stuff, so I enjoyed the presentation. I have no science background, so my questions are probably going to betray my ignorance about this. But I guess one of my questions would be, I have three. Like, my impression from talking to refuge managers and people in the community, like avian influenza is kind of always out there, right? Like, were you surprised to find it, or did you think you'd find more of it, or none at all? Well, did you have any kind of, like, maybe some sort of bias going in?
I kind of had the like I kind of knew that it's always out there, but the thing is, this method of detection hasn't really been tested widely to see if it's a very reliable method of seeing if it's always out there.
Okay.
So what this allows us to do is actually go through and test these ponds. So as Molly was saying, they go through and test sandy and coastal, but you also have other ponds that aren't tested as regularly, other places where you might not have, like the DNR is taking account of. So these smaller ponds are ones that we could actually hit with this methodology. We don't need to rely on 100 samples, but you can't go into like an apartment community and just start shooting the geese. So this allows us to go through and select these other ponds that are often more closely related and more closely associated with human activities than some of these other state parks and wildlife areas.
Yeah, the issue with it spreading is typically in concentrations, at least in migratory waterfowl, it's in areas where you have dense concentrations like snow geese are usually a prime, carrier every year. And it's real bad in states where there's not a lot of water, right, or the water's managed. Maybe, we're lucky on the East Coast, but I guess my question would be, let's say you could detect it in a refuge system somewhere, maybe on the West Coast where it's going to have a high concentration of birds. I assume there would be some management implications, right? Like if you found it before the migration, then maybe there would be a decision like, hey, is it better should we drain this place instead of having 50,000 snow geese show up and possibly get it? Or and that's something that I guess will probably come out, and then is there a way to treat the water and kill it if you detect it?
In terms of management implications, I can't really speak for that. My background is not in management.
It would just be interesting, right? If you knew in advance, hey, like this complex is teeming,
or it could allow people to, or could give the people who are making the management decisions, so like saying, yeah, we're seeing it in our water early, it might give them the chance to go through and up their
or increase the
screening
flow, or
and
do something different, to